Hello friends, myself Assistant Professor Neeta from RBNB College, Rampur. Today we are going to conduct our practical number 2nd from M.Sc Botany, paper number BOUT 234, that is seed science. Okay, then the aim of today's practical is study of grow out test, that is known as field test. Okay, in short we will call it GOT test. The principle of practical is the genetic purity of the given seed lot of release cultivars or the hybrid and extent to which submitted sample can be confirmed the prescribed standard in which the genetic purity test should be done or the genetic characteristic of the seeds can be calculated. Here we need some requirement and this practical is conducted into the field. That's why field is our first requirement. In case of seed we will use the paddy seed, which is commonly known as rice. We will use the paddy seeds and the quantity of the seed according to the choice of consumer or the plant experimental person. Then the selection of the field can be carried out through the basic concept. In case at the time of selection of seed we need to maintain the seed quality or the field quality. When we select the field, this time the field should be free from volunteer plants or unwanted plants, the noxious weed and the soil-borne diseases. We have to avoid this all concept. Then when we plant the paddy seeds or seedlings of the paddy, that time we need to keep in mind the definite distance between two rows, two plants or the two plots of the paddy seed. Here if we plant the seedling or the seed, that time we have to keep in mind the distance between two rows, that is a 6 centimeter. The plant to plant distance is the 15 centimeter and the space between rows, that means the space between or the distance between two rows should be 20 centimeter. And at the last, when we plot the variable variety of the seed or the species of paddy, that time the space between two different plots should be the 45 centimeter. Okay, this is the need at the time of plantation. Then for the genetic purity test or the field test, we have to observe this table. Firstly, when we plant the seedling or the seed, that time some seedling or the some plant should be off type, that means it does not form the flower or fruit, that is known as the permissible off type of seed, which is counted into the percentage, while the minimum genetic purity, that means the purity of out of 100% should be counted also into the percentage and the number of plant we planted. Here is the observation table. There are 4 observations and maximum and minimum genetic purity of this seed should be shown here. First one is the maximum permissible of type of seed percentage should be the 0.01%. Here minimum genetic purity should be the 99.9%, which is also into the percentage, and the number of plantation or the number of plants should be the 4000. Then second number, the maximum permissible of type of seed percentage should be the 0.20, where the minimum genetic purity should be 99.8, where in case of 0.01 it shows the 99.9% and here it decreases into the minimum genetic purity. That means the genetic purity will be the decrease when the maximum permissible of type of percentage should be increases, where the number of plants should be 2000. Then third number, if the maximum permissible of type of percentage should be the 0.30 and that time the minimum genetic purity should be the 99.7%, where the plant number should also be decreased, where 1350 plants are grown. And at the end, that means the last point, 0.50% of maximum permissible of type of plants or the percentage should shows the minimum genetic purity will be the 99.5% and the number of plants are 800. If other than the 4 types of readings or the observation, if the maximum permissible of type of seeds or the plantation or seedling shows the 1% or the above the 1%, maximum permissible of type of seed that shows the minimum genetic purity of 99.0 or it below. Okay, then the number of plantation or the number of seed that is purely genetically pure should be the 400. Okay, this is the observation table we should understand at the time of field test or at the time of grow out test. Then observation. When we test the genetic purity of any type of seed other than the paddy or rice seeds, we may take any one of our choice of seed. That time we have to observe the some characteristic at the time of observation. The first one observation will be the we have to observe the entire plant. Entire plant should be observed from the growing condition to the ripening condition. That means the formation of seedling toward the ripening condition. The second one observation is nothing but we have to observe the differential characteristics between two plants or the two seeds. That means the distinguishing study of two plants. Then second observation or next one observation is we have to confirm the plants are of type or not. Here in observation table, the first table or the first row is the maximum permissible of type of seed. In our plot there are many plants that forms the of type of plants. That means there is no formation of flower or fruit. That's why it is known as the of type of plant. So we have to confirm the plants are of type or not. This is the difference or the distinguishing characteristic between them. And the last one characteristic, that is the observation. We have to observe the entire morphological, that means the leaf auricle, collar region color, internode color and similar character like that we have to observe at the time of genetic purity test. Okay, if the observable characteristics are normal, then we can say that the plant we planted is the genetically pure or it formed the pure seed lot. Okay, thank you.